Thank you very much. It's a great honor for me to be here and to see the auditorium full of people. I want to thank especially the ones that organized this school, advanced school. I never did myself one, but I imagine it must be a hell lot of work. So Cristina, thank you very much, and all the organizing committee, Cristina, Mesquita, Banhato, Morayama, Inada, and Prata Vieira. So you guys did a wonderful job. Thank you very much. So since it's advanced school, I propose to give a lecture in which we try to give one whole message. And the idea is to at least first define what is these content-dependent reactions and why we think they are so important to real outcome in PDT. So we are talking about this business of treating disease, mostly proliferative disease, cancers or infections with light, using a pigment that absorbs light and do a lot of chemistry and biology here, and these outcomes in a treatment of the disease. The key questions I start to ask when I start doing research as an independent person 20 years ago is exactly how can we understand the mechanism? And if we do understand the mechanism, how we can optimize the process. And so since then, I've collaborated with some medical doctors, especially a friend of mine called Tardivo, and he has treated hundreds and hundreds of patients of different diseases using a very simple commercial drug, which is methylene blue. Many people here use this drug, but he makes kind of a miracle of this. So this is a process that works. Okay? The problem is sometimes the outcome is not as we think it is, and still it's very empirical. Since we have many factors involved, any treatment, medical treatment, is kind of empirical. You have a drug, you don't know what the concentration the drug reaches the tissue, disease tissue. Each of us has a different environment of the disease. And on PDT, besides all of that, you still have the light. So there are many factors, and there is empirical, but if we do not understand the mechanism well, so we cannot propose evolution. So the fact is that it works, but if you ask a medical doctor in any specialty except dermatology or ophthalmology, they probably don't know. They never heard about it. Oncology, they think it's kind of exotic, so you use light. So in my view, a lot of is because people don't really understand the mechanism, and that's why we try to do that. So I just give you a few hints of the work of this friend of mine. Now he's very involved in treating the diabetic foot. So it's a disease that attacks the vasculature of the foot. Person becomes infected with anything happening in the toe. So it can have also myelitis, and then the person can lose this. And what Tardivo does is treat these patients every week. And so this is the PDT group. He just lost one patient because he was traveling abroad. This patient went to the hospital and got the toe cut. And the control group, all of the patients lost some part. So what does he do with PDT? He treats the bacterial infection, for sure, but also there may be some other effects. I mean, look at the bone in X-ray before treatment. So you could not even see the contrast. And after PDT, the bone is a bit distorted, but this person was not walking, and this one is freely walking. So it's a beautiful work. It's basically the reverse what A.V. Dor mentioned to you, because he was shutting down blood flux, and here you need oxygen to treat this disease. So he always needs to find ways to make PDT better in order to make more oxygen going through. So what happens with this when you form an excited state? You form a species that's more reactive. Usually, if this species is not attached to a target, so it needs some time to encounter by diffusion some reactant. So triplets are usually considered to be the main responsible for the effect of PDT, because they have open ground state, so it can be a very strong oxidant species, and you have an excited state with electron more, so it can be a very good reductant species. So redox properties of excited state are both a lot better than the ground state. They are better oxidant, they are better reductant species, but also the triplet has special reactions with oxygen, for instance. It reacts with oxygen and forms oxygen single oxygen. I'll mention more about that, but also does a lot of redox chemistry. So this is what we know about the mechanism. Is focused on the properties of the photosensitizer by itself. So we call these two different types of mechanisms, type 1 and type 2. The type 2 is an energy transfer reaction. The triplet species react with oxygen. This is the one we breathe. You see it's a triplet species in the ground state. So that's why we do not burn when we breathe, because you see oxygen has 2 electrons that are in 2 orbitals of the same energy, but they are parallel electrons. So if you want this oxygen to add to the double bond has 2 electrons that are with inverted spins. So you can never fit the 2 electrons here. It needs to be a spin inversion. However, single oxygen, it has an open orbital here. So it's a very strong oxidant, and there is no problem of spin to react. So it adds to the double bond. And also you have possibilities to react, the biomolecule react with the triplets of the singlet and form radicals, and from here you have radical chain reaction progressing. So that's what we know. That's what has been happening in the development of new photosensitizers in the PDT community for decades. So people are trying to go for better absorption in the red. So if this pointer would be red, you'd see your finger red. If it's green, it doesn't penetrate too much. So this is evolution from porphyrin to chlorine, bacterial chlorine. Avigdor mentioned the molecule that he has is a bacterial chlorine. I mentioned some data with a chlorine. So the thing is, the spectrum you see goes to the red. The other thing that people have developed during the years is to make molecules that make a lot of single oxygen or other reactive oxygen species. So this is basically the 2 factors that are being used to develop new drugs. And so since this is a lecture, I want to interact. If you guys don't respond, I was stuck here and we stay in this slide for many minutes. So let me ask you in the audience, who may think of other factors besides these 2? I mean, of course, the molecule has to make some reactive oxygen species. Otherwise, there's no sense of talking about photodynamic therapy. But is this really the factor that we should modify in the molecule to make it a better photosensitizer? Penetration of light is important as far as deeper you want to go. There is no way you want to avoid that. If you want to treat a deeper tissue, you need more light. But today you can put light anywhere. With proper fibers, you can really treat any site putting light right there. So are these the 2 factors that are the major important ones? That's the research we have been doing. But before I show it, I want you to suggest all the possibilities. What other factors may affect a photosensitizer? Let's suppose you are a PI now. You are the investigator. You want to run a research line. What do you propose? What else should we test? So that's a good point. So who is Mark you mentioned? No, you guys. Okay. So you want a drug that is not toxic in the dark. You have a good point. Many of these drugs are toxic in the dark, a little bit, but if you put light, it becomes a lot more toxic. But of course, this ratio is important. The toxicity compared the dark with the light toxicity. But I mean, talking about properties of the molecule itself. Yes? I couldn't hear you, sorry. Can you speak louder? Okay, so you're talking about 2 different things, right? Huh? He mentioned, so in terms of targeting, we can think of drugs that will target tumors by the APR effect, so it depends on the size. And also, another thing you mentioned, drugs that will enter the cell and target organelles. Yes, I like your idea. I do not work with PR, I do not work with physiology of drug delivery, but I do work with intracellular targeting, and tomorrow I have a full lecture to deal with that. But thank you very much. That's a good idea. Yeah. Two other factors that have no relation with this. So if you take a drug that is not very efficient in terms of single action and put it inside the mitochondria, how many photons you need to absorb for this drug to kill the cell? Not many. That's a good answer. That's true. Other suggestions? Okay, let's go progress a little bit. So that's the question we put ourselves to some time ago. So how did we answer this question? We have many years of research here in which we compare molecules that have the same center of the molecule. It's the same chromophore, but it's different peripheral groups so that the interaction with the cell or with the membranes, with the proteins are different. And they are basically controlled by how we make these peripheral groups. And there are two important features that I want to discuss with you in these two lectures that are, in my view, a lot more important than the yield of single action. Okay? And one of them is amphiphilicity, how well these molecules bind to the membranes. And I will discuss this today. Why is this important and how important this is? And the other one is cell localization. Our friend already mentioned this could be a good target, and you're right. And tomorrow we'll discuss more about that. So how about amphiphilicity? If you look at the literature, look at the big bucks in PDT, molecules that really made a lot of money. This hematoporphyrin derivative, it is a kind of amphiphilic molecule. It's not very soluble. We don't know exactly the structure, but it stuck to the cell tissues, becomes photoactive for months. And then, so that's one of the main problems of these molecules. This binds too much. So, but the other two molecules, like verteporphyrins, and the one we mentioned, we heard about today, they are kind of amphiphilic, and they bind, they interact with biomolecules. And when they do, this may shift a lot the photochemistry. And if you look another details of the literature in people that do research on this area, and they do basically what I used to do also, what I do myself. We compare series of molecules, and we vary the structure of this molecule and see which ones are more effective. And I could never find one paper that would find the amphiphilic molecule less efficient in any model you may think. Take this work of Jory. He's a disease guy. Unfortunately, he died, but he did a lot of research on this. And he used amphiphilic molecules to treat cells, to treat bugs, to treat larvae, and all of when you compare the efficiency of these molecules, the amphiphilics were more efficient. Whole animals treated. I mean, these people use a lot of cell studies, and they could see the efficiency as a function of log dP. Durantini is a guy from Argentina that basically makes synthesis. He compares efficiency of antimicrobial therapy in a series of molecules. Always, the amphiphilic molecules are more efficient. This is a very good trend. So we did some work also in collaboration with Mark Wainwright. He's here. I hope he can also agree with me. Otherwise, you can talk to him. He's there. So not only for porphyrins or ring systems like this, phenothiazinium ions also work like that. So why is it so important to have a molecule physically attached to the membrane so that it becomes more efficient? That's the question I want to ask today. But before that, I want to show two data of my lab. So look at this series. So this is the work of Pavani. So she was a postdoc in my lab, and she compared the efficient series of porphyrins, varying these groups here. And you see, this is the amount of single action. All of them make the same amount of single action, but the efficiency of cell killing varies a lot, from almost nothing to almost 100% in the conditions that we treated. But when we compare now as a function of membrane binding efficiency, now we could select in two groups. The ones that bind more to the membranes are the ones that are more efficient compared with these two. So another example, also another series of porphyrins. You see there are several charges here, but if we compare these two here, you see these two both have two positively charged on them, on these molecules. But this one is symmetric molecule, so it's a trans isomer, so it means that it's not really amphiphilic, but this is a cis isomer, it's very amphiphilic. And if you compare the efficiency of these two, look at this is destroying red blood cells, and you see this one, it takes a hell lot of time to irradiate, and this, you start to irradiate in a few minutes, they're all destroyed. And this one is in between, but it's a lot less efficient than the cis isomers. So this guy was able to put this in the computer and understand why, in terms of interaction, this is more efficient. So, but that's the main question today. So why membrane binding of photosensitizers is so important to cause cell death and the photoactivation. Now we stop here. I want to hear again. Yes, please. Yes, so you're talking about if the molecule is physically bound, it can cause lipid peroxidation, and then this is very important to membrane destruction. Is someone else raise your hands? Is it the same explanation?
No, I think that's because the lifetime of the secret oxygen is very fast, and then it has to be near what you want to kill.
Okay, do you think, I mean, your answer is correct, of course. I mean, there is more membrane oxidation, lipid oxidation, but why you need to be physically attached to have physical membrane oxidation? And he started to go into that direction, but this is not correct. I will show to you why. But do you have any other? Yeah, I mean, the biology is very rich. I mean, as soon as you damage a mitochondrial membrane or reticulum endoplasmatic membrane, all of this can happen. But let's focus on the photochemistry of the photosensitizer, photosensitization in the membrane. Why do we need to physically attach it? So any other idea here? So you're thinking the cell may have an antioxidant that does not protect against the membrane? Because, I mean...
Maybe it is more inside the cells, but not on the outer part of it.
Well, a lot of antioxidant actually focus in the membrane. That is the truth. So, yes? This may occur, and I'm planning to discuss this tomorrow, yes. So let's progress a little bit. Concerning the single oxygen, so if we do not really calculate this diffusion of and reaction of this molecule, we may think, and we did that for some years. So the molecule has to be physically attached, because in the membrane there is more oxygen. Actually, the concentration of oxygen there is higher, that's true. And so if you have the molecule here, you have more single oxygen here. But that's not true. The real fact is that it doesn't matter if the molecule is here, the photosensitizer, or there, if you apply equations of diffusion and reaction, and you can measure that. You can have conditions in single oxygen is done here or there. The single oxygen amount will not be higher here. And the reason is the following. Almost all the single oxygen that will be generated, if the molecules physically attached here, will go out of the membrane. That's the fact. You can never measure lifetime of single oxygen within the membrane. Using the best equipment that is available. What you can do is actually fit the data by the going out, diffusion out of the single oxygen from the membrane. That explains a little bit. But statistically, if you have this molecule within this diffusion pathway, the amount of single oxygen that will reach the membrane is the same. Okay? So that's the first problem with this. So why do we need the molecule to be physically attached if the amount of single oxygen is basically the same? If the molecule is here or there? So who can help me now? Yes? You want to help me? Huh? Yeah, so it seems that contact is really important. So, and I don't know if you realize in the lecture in the morning by F. Victor, the molecule that he used physically attached to the protein. It goes inside the pocket of the albumin, right? So in order to really understand what happens when photosensitized oxidations occur within the membrane, approach the membrane, we have started a research line in collaboration with a professor, colleague of mine from physics in São Paulo. She's Rosângela Itri. We have done a lot of research together. I will simplify it in 2 slides. So one slide that I mentioned to you is we thought this would be a very powerful photosensitizer. So this was designed by a previous student of mine at Jaci. This is a chlorine, and so he made a chlorine with a tail. So it stuck to this is anchored to the membrane. So we thought when we synthesize this, so this is going to be the best photosensitizer ever to destroy the membrane because it's physically attached and doesn't aggregate. So there is this molecule here has an L-shaped structure. So if you can check in JOC, you can put millimole of this molecule, as much as you want. It doesn't aggregate. Okay, so we thought this is the best photosensitizer. But then we start to do photosensitization in the membranes, and actually this molecule doesn't destroy the membrane. It just makes the membrane gain more area. So you can pull it with a special device and measure how much area it's gained when you have oxidation of the lipids. And basically what we determine is that this molecule only makes one reaction. It only makes single oxygen within the membrane, but the single oxygen attacks the double bond and makes a hydroperoxide. So think of a lipid, a membrane full of lipids stuck there, and when you put a hydroperoxide here, this lipid has a natural tendency to do like that. So you increase the area per lipids, okay? And we can measure that. We can quantify. In this paper, published in Soft Matter, we actually were able to quantify what is the yield of reaction of single oxygen with the double bond based on the amount of area increase. We are also able to see maximum expansion. A lipid that has 2 double bonds, the DOPC, expand maximum 20%. So this is an expansion of the lipid. POPC that has 1 double bond less than that. But we also measured the stretching module. Is it easier for the membrane to stretch when it's oxidized compared to when it's not oxidized? Yes, it's a lot easier. From 200 millinewtons per meter, that's the force it takes to stretch the membrane. When you make it full 100% hydroperoxide, it goes to 50. So 4 times more flexible. So single oxygen does make a lot of things in the membrane, but does not destroy the membrane by itself. Here we can make 100% hydroperoxide membrane and doesn't leak. You see some of these points here were done in situ for oxidation. Some of them were prepared by isolating this molecule and preparing vesicles. Okay, so what other things hydroperoxide and lipid single oxygen does with the membranes? The other thing is work that we have done some work before, and the lady that's here, it's called Suboni. She's over there. She can explain better to you this last step, but I will go quickly. So what happens is if you have a membrane that has a mix of lipids, and these lipids are there balanced, they are mixing themselves in the membrane. If you start to form lipid hydroperoxide, you start to demix this. So you start to form some cholesterol domains, and this you can see. So, and what Tayana and Suboni, what she's here, she proposed that in the first step, when you form only hydroperoxides, and then you have formation of domains, because you have this, this force that's called hydrophobic mismatch, lipids with different size, different polarity, and when you form hydroperoxide, you increase this, so you form domains. But also if you progress it, and then you start to cut the chain, and you form now other problems, leak in membrane and so on. But concerning membrane leakage, what really is important? So I mentioned to you that we can fool 100% hydrogen peroxide in the membrane and it doesn't leak. So what really makes it leak? So we can also make experiments in the lab using this system of grooves, which are giant unilamellar vesicles. We see the membrane in the microscope and we monitor the reactions. We can see that it depends on the photosensitizer, it destroys the membrane. So you see, what starts like this, and after reaction goes like that, complete destruction. So at this time, this is some time ago, we were able to detect some of the short chains amphiphilics. They go to the surface, and we can detect a decrease in surface tension. But we didn't know exactly what was happening. And so what can happen here? Two main reactions. One of them is based on single oxygen, and you form the hydroperoxide, and we know the consequence of this. But the other one is a radical chain reaction and end up cutting the chain. So it's very likely that the reason why you need to physically attach the chromophore to the membrane is because it needs this type of reactions that are not occurring because of single oxygen. So as a scientist, you have to understand that we make hypotheses. So I knew the response before I started the paper I'm going to show now. I had a very strong feeling that this was necessary completely for the membrane leak, but how can I prove that? How can I make everyone knows about it? How can I make people believe me? That's a very, very important step in the life of a scientist. We always have ideas, but if we do not work hard to make other people understand and believe, it's just an idea. So this is a very clear idea, right? You think when I've done this work, it's finished. No, it started. This project of this lady, Isabel Bacelar, she was a PhD student, working also in partnership with Mike Wainwright. He has very, very nice photosensitizers, so this is one of his creation. He gave several names. We used this when it was DO15, but now the name is S137. People are using here, so I saw one of the lectures. And so these two molecules are very similar, right? The chromophore is the same, but this is a bit different. Mark make it become a lot more efficient, bound to the membrane, but also avoiding aggregation. And what Isabel did was to do the leakage experiments and do the physics so that she understands all the pathways that were happening and measure the reactions products that were occurring within the membrane. And so the physics, so she make a system in such a way that either methylene blue or DO15 produce basically the same amount of single oxygen reaching the membrane. We can do that, okay? That's easy. That's simple equations we can use. We can test that. So basically, both dyes generate the same amount of single oxygen reaching the membrane. This is about 150 per second, okay? The difference that the DO15 is a lot more efficient in terms of membrane binding. So the KB is 265 compared to 13. So here in 1 to 7 binds to the membrane, here is 1 to 15, 150. So this is more efficient in binding the membrane. Also, another thing, this molecule goes deeper in the membrane. It reaches closer to the double bond compared to the ones. So the ones that are bound, these are a lot more bound, but also bind deeper in the membrane, getting close to the double bond. So if you measure the efficiency of a possible electron transfer reaction between DO15 and this double bond is around 30 times higher efficient compared to methylene blue, okay? And when she does leaking experiment, how this leaking experiment is done? So you take a liposome and you put molecules fluorescent inside the liposome. But they are in such a concentration that are self-quenched. So we don't see the fluorescence of these molecules that are inside the liposome. And when the liposome leaks, we start to see fluorescence. So it's a very classical way to to study membrane leaking. So you see, only at these conditions she prepared, only the DO15 was leaking the membrane. Methylene blue was done any leaking at all, okay? And so she went to these conditions and started to see the products of reaction, what was accumulating in both cases. And both cases work, so she prepared of these compounds, a very demanding project in terms of making compounds, analyzing the mass spec and so on. So what she defined in the end is that, I mean, this is the date of leaking, only DO15 was leaking. And you see here the products of oxidation. Both of them were making hydroperoxide. So this is hydroperoxide by DO15, and this is hydroperoxide by methylene blue. This is more, because it is more efficient, but you see both generates single oxygen and hydroperoxide. The other thing interesting is that the yield of alcohol to ketone were basically the same only for DO15. We couldn't see that for the other compound. So this is an idea that we do have some mechanism called the Russell mechanism for oxidation. But the most important aspect is perhaps the fact that she was able to detect aldehydes. And always, when she was able to detect aldehydes, the membrane was leaking. So this is the data for DO15. So this is the level of aldehydes when the membrane was leaking. And methylene blue was a very poor compound to leak, but if she allows it to react for hours, okay, so a lot more time, this is 20 minutes, this is 3 or 4 hours. I don't remember exactly, but it's several hours. So she starts to see leakage. At the same time, she starts to see aldehydes. So this is a proof that in order for the membrane to leak, it needs to accumulate aldehydes. So in how the aldehydes are formed. So now comes the step in which we had to understand how the aldehydes are formed to really understand how these two molecules work differently. So the photosensitizer makes single oxygen. Single oxygen does react with the lipid, makes hydroperoxide. But then there may be a lot of reactions here within the membrane. What are the reactions that are really leading to aldehydes that cause membrane permealization? So the first thing she did was to compare the reaction of the photosensitizer with the lipids themselves. And you see that here, if the lipid have no double bond, there is no reaction. If it has one double bond, if it has two double bonds. So the efficiency of the reaction of the photosensitizer with the double bond increase, okay? And this is something showing that the photosensitizer is here, and it's reacting with the double bond. Does the photosensitizer react with the hydroperoxide? Because another possibility to form aldehydes is that the photosensitizer reacts directly with the hydroperoxide. And yes, indeed, we do have an evidence. This is still an indirect evidence, but if you do the efficiency of a probe, which is a probe that detects these guys here, peroxyl and alkoxyl within the membrane, the efficiency of this probe is a lot higher for hydrogen than for D2O. This is the reverse that was expected based on single oxygen, because single oxygen has a very strong lifetime in D2O. But this is also compatible with the fact that this exchange, so if you have D2O, it's more difficult to remove this deuterium compared with hydrogen, so we can explain that by this. And so I'll go a bit into the end point. So the other thing that we need to discuss here is the mechanism by which alkoxide leads to aldehydes. There is a mechanism called beta scission, in which the alkoxide has a break here, and you form the aldehydes, and you break this chain. So in here, you form a lipid truncated aldehyde. But there is another mechanism possible that people have cited in the literature called Hock cleavage. In this mechanism, you actually do not need to arrive here to have the cleavage of the chain. So if you have a hydroxyl close to a double bond, and this happens all the time when you do in reaction forming single oxygen with the double bond, you would also do this. But actually, nobody ever saw this reaction happening in lipids. They observed this in polymers. And one paper in cholesterol, but never in phospholipids. And then we tried, can we see this reaction in phospholipids? And never. So you can make a membrane that is full of hydroperoxide, and this one, and you can go pH down up to 3.5, and the membrane is still intact. So this reaction depends on acidic catalysis, does not really happen within the membrane. And so this explains to us what happens. single oxygen is very important to make the membrane more flexible, to gain area, to have domain formation. So this may be very important for signaling. But if you want the membrane to be destroyed, you need the photosensitizer to come here and to react within the membrane to form aldehydes that will get to membrane permeabilization. So that's the key point here. You need the triplet close to the double bond. This is a contact-dependent reaction. So now we prove to the society this is important to destroy the membrane, and that's the mechanism, okay? It took 4 years for Isabel to finish this, and a lot of partnerships for several people. Thank you, Mark, very much for being such a good partner also. So I hope you agree with me here. Does anybody have a point that I should consider that's against this hypothesis? Do you agree that this has been proven at least for membranes? Okay, so one thing that happens is when you have this electron transfer reaction between the membrane and the photosensitizer, there is a very large chance of having photo-bleaching, okay? So this is the fact. You have electron coming to the membrane, this distorts the electron orbitals, and then the chance of this molecule photo-bleach is very high. Citing A. Vigdor again, he mentioned that when the chromophore is in solution, there is a lot of photobleaching because of this electron transfer reaction. Within the protein, somehow it doesn't happen. He's still understanding the mechanism, but very likely there is some way that the protein feeds the electron back. So the photosensitizer loses electron, and then the protein feeds it back. So there's a refurnishing mechanism. But before going to that, so I think this is very important for new PDT development. That's my point I want to reach in the end. But before we get there, I want to ask yourself, do you think there may be a relationship between photobleaching and efficiency of membrane leakage? Who can help me that? Do you understand the question? Let's suppose there are two molecules. One gets photobleached within the membrane, and the one does not. Which one is more efficient to cause membrane damage, to leak the membrane? So who wants to help me here? I went too fast? The one that does not photobleach is the one that will make the membrane leak faster, right? This is common sense, yeah. I mean, if photobleach, I'm not saying that we should all use molecules that photobleach very quickly, but I want to prove a concept that perhaps your hypothesis is not completely correct. Okay, because that's the common sense. I mean, if the molecule vanishes, there is no absorption, there is no reaction. But is that really true for membrane leakage? So that's the question I want to. So who answered this question is this guy. He's Tiago. He also was a postdoc. Now he's a professor in Juiz de Fora. And he is an organic chemist guy. He made a lot of these porphyrazines molecules. You see, different to porphyrins, these molecules have nitrogen in the ring, okay? So this makes a little bit different in terms of photochemistry, especially in terms of redox chemistry. So these molecules can stabilize radical species which cannot be occurring in porphyrins. And he made a lot of them so that the peripheral groups are different, and we can tune a little bit the redox properties of these molecules. And so I will not go into very detail here, but what he shows that this molecule photobleached. Actually, we started this work to prove the hypothesis that if the molecule is able to stand photobleach, is a more efficient molecule. And this is, if you take new pigments that are coming to market, they are fluorinated here to remove electron from the chain and then avoid the reaction with single oxygen. Because some of the chromophores are killed by single oxygen because they have a lot of electron density. If you remove electron density, and then you avoid the reaction with single oxygen. But in this case, these molecules do not react with single oxygen, any of them. And the reason is because of the porphyrazines do not have redox properties to be oxidized by single oxygen. So they are stable under single oxygen. But they do react with solvents. You see, it depends on the solvent you put. If you put in pyridine, they vanish very quickly. And depending also on the peripheral group, there are big changes. So this trifluorocarbon compound, it does photobleach a lot faster than the fluorinate only, okay? And this can be explained by the properties of these two ligands. I'm not gonna go into detail here, okay? But these ligands stabilize better radical anion species compared with that one. So removes better electron. Fluor ends up putting some electron back in the ring. And so when we do this photochemistry, we put in DPR, we see a clear picture here of a long-lived radical anion species. So what happened is that this molecule is removing electron from the solvent, okay? And if we put this molecule within the membrane, it will take electrons from the double bond. And this is the case. I mean, look at the photobleaching efficiency. The MPC basically does not have photobleaching. There is no double bond, with one double bond, with two double bonds. So the molecule bleaches faster if they're interacting with the double bond. So taking electron from the double bond and getting photobleached. But now comes the data I want to discuss with you. So look at this molecule. It's the one that's more stable. It's less photobleached. And look what it does to a membrane. It does leak the membrane, but in a different rate. This is the one that photobleaches faster. It bleaches the membrane in a faster rate. So that goes in the same direction. In order to have a membrane leak, you need to have a radical chain reaction. And who starts this radical chain reaction? The chromophore, the pigment, can take electron from the double bond, or can give electron to the double bond. In this case, take electron from the double bond, and then start the chain reaction. And this is, but when it does so, it photobleaches. So the take-home message here is that you guys that want to develop new photosensitizer, why not thinking in a way to make it contact reactions and refurnish it? So Avogadro is doing that already, but we can think of other things, other nanoparticles, other proteins, other ways to have it contact-dependent reaction, then have it avoid the photobleaching. So I want also to discuss if we should, when we write these Jablonski diagrams, we give basically the same importance to these diffusion species, and actually we don't almost not mention this. And I think we should start changing the way we write this. So you should give more emphasis to these contact-dependent steps, which are really the most important to make damage in biological tissues. So, again, the question, why membrane binding is so important? Who can answer to me? If you don't answer, I'm stopping the lecture here. You're not going to see the rest, which is nice also, so I apologize for that. Who is going to help me here? Nobody? You don't want to see the end? Who wants to help me? Why membrane binding or photosensitizers? I'm sorry for the English, so it's a bit of a problem here, but okay, let's take cell death out and put membrane damage. Perhaps I should, yes.
Said many about lipid peroxidation, but maybe influence of reacting oxygen species on membrane protein-like receptors. It's more good mechanism for cell death, because peroxide modification of the protein can lead to activation of different pathway, I think.
Well, thank you. I think I understand your idea, and it's important. And the question, I think, was a bit driven to this answer, actually. But what I want to hear is photosensitizers to really cause irreversible damage in the membranes. They need to be physically attached to it, because the key reaction is an electron transfer reaction with the double bond. Okay? And this is maybe occurring with photosensitizers that are bound to protein, bound to DNA, and so on. So that's the message, the take-home message I would like you to take. And of course, I want to discuss if you have any questions concerning this. Saying that, I'm not saying single oxygen is not important. In my view, it's becoming clear and more clear every day that it's very important for signaling. There was a paper coming out in Nature a month ago. I read to you, single molar oxygen regulates vascular tone in blood pressure in inflammation. Okay? And part of this data was obtained in our lab. I'm not a co-author, but a colleague of mine in the department is. And so we have in our department a very, very important facility to measure single oxygen. So I will show you very quickly 3 equipments. One is a spectrometer, time-resolved spectrometer, so we excite the samples with lasers. We have a monochromator so that we can have the spectra of emission and also the lifetime. We can measure how quickly vanish in water, and if you put something that will react with it, we can measure the rate of reaction. We are now working in the imaging system, so this would be single oxygen phosphorescence imaging in a partnership with PicoQuant. We designed this. The issue here is very important because single oxygen is a diffusion species and an emission species. So within the confocal volume, always you have escaping molecules. So there's a big mathematics coming to play a role here, but we can see if you take beads that are in water, D2O, or air, we see a clear difference in lifetime. You see here, in this case, lifetime. So we're basically doing fluorescence, but this is phosphorescence emission lifetime measurements in the microscope. And Caetano, the guy that's here, he's always playing in that equipment. He still haven't published anything on this because he's now a company owner, but I hope he gets these images published very soon. The last equipment I want to mention is the one that we just developed. Actually, this was developed by people from Humboldt University, Beata Roder, Stefan, and Jan. Jan spent 2 years in the lab, and we bought part of this equipment. They have a startup company also. And you see now, look at the lifetime I have in my equipment, the old one. I cannot see the rays of single oxygen generation. So I cannot see the triplet being suppressed by oxygen to form single oxygen. But look what I have now. Look here. So I can study triplet lifetimes now on this equipment. So I finished. Thank you very much. And so I hope we can discuss a bit this issue. This is it's an old picture, but that's one I have. Diana is here already, but she's no longer in the lab. She's still interacting. So thank you very much.
Thank you, Professor Baptista. It was a wonderful talk. Do we have questions for him?
Yeah, it's really amazing talk, and I really like the membrane bounded question. So I want to have a high-level question, right?
Okay.
So if single oxygen binding the membrane is really what caused the cell damage, so would you consider with the Hidetaka Kobayashi's photoimmune therapy, he's claiming all his PDT works because of the membrane, bursting of the membrane, so that's one. The second is all the historical literature showing the membrane-bound photosynthesizer is magnitude order of less effective than the mitochondria and the other organelles. So what do you think about that?
Yeah, so the first question I want to talk to you in more details because I think it's a very nice question, but I cannot comment on public because I don't know all the details I should, but I would love to discuss with you. You're right. I mean, I'm not saying this is cytoplasmic membrane, but in my view, of course, if you do damage in the internal membranes, organelles, you have a lot more efficient. And we have data in the lab, several-fold increase in efficiency if you target or lysosome or mitochondria. But both of them, you're targeting the membrane of these organelles. And so still, this type of understanding helps. So thank you very much. The guy had a question. Yes.
Thank you very much for your talk. I would like to know, there is a group at the University of Oslo led by Professor Christian Berg, who has developed a technology that they call photochemical internalization.
Yeah.
And that mainly results from the fact that the photosensitizer mainly accumulate into the membrane of the lysosomes.
Yeah.
So I would like to know, like, is this technology the same as you're applying, like with an amphiphilic photosensitizer binding to the membrane, but damaging it either or making it porous enough for?
No. Yeah, I know the concept. I know well people that have developed. I know people that are trying to show that this really plays a role. And I can have my own view on this, okay? Yes, the photosensitizers are amphiphilic. They are the ones that bind to the lysosome. They are used in a very low concentration. So the concept says the following. So if you damage the lysosome, whatever is inside is released, and then you can really treat, it's an internal drug delivery system. You avoid the endocytosis, fusion with the lysosome, and then if there is a protein, it will be digested, okay? I think this has been partially proved in some case, but it has been very difficult, challenging to really make this in clinics. And in my view, if you damage lysosome, and we do publish, I published a paper this year, there are so many other things happening in the cell. So it's a very simplistic view of what happens, because lysosome has many, many functions. And so damaging in the lysosome membrane is not really opening a bag. The cell responds in many different ways. It can be, for instance, blocking autophagy, and this kills the cell. So is this really the release of the material that's killing the cell, or is it blocking autophagy? This has not been proven yet.
Okay, sorry, I would like to ask. Then my second question is, you've said that using this PDT and the singlet oxygen, we can have the expansion of the membrane. Can this expansion be large enough to make the membrane porous and therefore releasing any small molecule instead of damaging like they're doing, like just making the membrane porous?
No, with lipids. We cannot say anything yet if there are proteins within the membrane. So the proteins may, if the leakage becomes, if the membrane becomes fluid, as I mentioned to you, the protein may feel a different environment and may start to leak. I don't know that. But if they're full lipid membrane, the hydroperoxidation does not make it leak, okay? We measure many times with many lipids, even PUFA lipids, okay? We can make a membrane that is 100% PUFA, 100% PUFA hydroperoxide, and these membranes do not leak. Okay, the hydroperoxide is not making the membrane leak.
Thank you, Professor. This course will continue tomorrow. Let's thank him again, please.
Thank you.